All right, so today I'm going to continue the topic. Started from last week where we presented some pipelines to extract connectivity, different types of connectivity from the brain, functional connectivity, structural connectivity, and you have seen some matrix, right? So today I'm going to present topic called modeling human brain connectivity from discrete networks to continuous functions. And the reason I present this is because last time I think we introduced two different pipelines. One is ROI-based discrete isolation, and another one is some continuous functions on the brain. So I'll try to introduce, methodology to work with these two different type of connectivities and talk about pros and cons for this different connectome representation. Okay. So again, human brain connectome data, you've seen this slide before. It's gonna be the data we're gonna use most of the time in the paper I present in the following slides. The reason is the Human Connectome project can be easily accessed through the internet. You don't need to get some IRB approval, etc., to access the Human Connectome project. And to remind you, this is tractography you can build from a diffusion, person's diffusion MR data. And it contains 1 million fiber curves connecting different brain regions. And what we try to do is we try to analyze connectivity generated by this this set of fiber curves. So one way to do it is to analyze that object, or we call structure connectivity, as discrete network data. So how to get this discrete network data? Let's see if I can make annotations here. So how to analyze the complicated objects generated from tractographic algorithms as connectivity matrix? What we can do is we can segment the brain cortical surface into different brain regions, right? This is so-called ROI1, this is so-called ROI2, and you have many of them, right? So in general, let's say 168 of them, ROI 168. So if you treat each of those brain regions or cortical regions as one regions of interest, we can look at the connectivity between each pair of brain regions. Okay, so this pair of brain regions connected like this way, this pair is connected in another way, and then we have another connection. So if you count how many of fibers, counting is just one feature, we're going to talk about it later. If you count how many fiber curves connecting a pair of brain regions, what you're going to get is a connectivity matrix, right? So this is RI1. This is RI1. This is RI2. Each rule describes one RI's connection to the rest of RI's. Okay, so we can get this connectivity matrix. And depending on different features, we extract, say, the number of count between two regions. We can get that matrix, or the diffusion profiles like a fractional anisotropy, FA value, MD value, and so on. You can get a mean value to describe that fiber tract. So you can get those connectivity matrices. Okay. So this is the discrete way of analyzing brain connectivity. In the SBCI pipeline we introduced last week, it contains a small sub-pipeline called population-based structure connectivity. It extracts those ROI-based connectivities based on two postulations. One is dead scan, another one is destrios. The procedure is like this. You got a stratigraphy result, contains millions of fiber curves. And you have brain populations, and then you look at two regions of brain, and you extract the fiber curves connecting them. Because the stratigraphy algorithm can produce lots of errors, we have to somehow analyze the track connectivity, the tracks first, and remove some outlier fiber tracks. And then each fiber track can connect multiple ROIs. So there's a cutting procedure. Let's say this track passing 3 ROIs. We're gonna cut this track into 3 pieces. One connecting this two, one connecting this two, and one connecting the further one. So using those kind of data cleaning strategy, and then we can build a matrix containing fiber curves, ROI1 to ROI2, ROI3 to ROI1, and so on. So each cell contains fiber curves connecting different brain regions. And then from this fiber curve, and you can extract the features to describe each connection. So when I say features, it can be weighted feature to say how many fibers are connecting, what's the average FA value, and so on. This is a weighted network. And then we also can binarize whether we can look at whether two regions are connected by fibers or not. You can get binary level connectivity. And going back to the original level, you have those fiber curves, and you have this matrix. Each matrix contains the fiber curves. And it's pretty comprehensive data from complex to simple. In terms of the feature you can extract for a pair of connectivities, let's say this is one ROI, that's another ROI. There's a fiber curve connecting these two ROIs, and we want to extract a feature to describe this connectivity. And what kind of feature we can extract? We can extract many features. For example, diffusion-related features. Fractional anisotropy, mean diffusivity, and also some matrix you can extract from ODF functions, the generalized anisotropy, apparent fiber density, and so on. So lots of features can be extracted to describe the connectivity between two regions of interest. And also there are some geometry-related features like the length of the fiber, how long of this fiber in terms of average connecting to ROIs, number of clusters. It means how different pathway, how many different pathways connecting the two ROIs. And also the average deviation from the mean fiber, that is the standard deviation of those fiber curves. Okay. We can also extract topological features like persistent homology and so on. So many features to describe the connection. And of course, the endpoint is a very important, can generate very important features, like how the fibers connecting those regions of interest. Those regions of interest are actually gray matters. Gray matters are the computing units in our brain, right? How those fibers connect in those computing units is really important. So the endpoint also generates some important feature. One feature is fiber count. How many fibers length, how many fiber lengths on the ROI. Okay, that's one feature, and also connected the surface area. We can draw a small circle for each endpoint to this region, and then we calculate, the total area covered by this fiber ending points. If we say the ending point has a small area. Okay, and then also the weighted connected surface area, which is some kind of ratio related to how many fibers. So if you extract those features, what you end up with is many different type of connectivity features, right? For each brain, we can have the curve in the log scale describing how many fiber curves between two regions of interest. We can have average fiber length, where you can see that between hemisphere the length is pretty big. Within hemisphere the length is relatively short. And we can have maximum FA, mean FA, connected surface area, which is the feature we just introduced related to the endpoints. So you got many features. If you put those together, you stack those together, each feature is a 2D matrix. If you stack them together, you get a 3D matrix, right? If you stack them together, you get how many? Let's say M of them, because we extract the M. So in this case, we have M equal to 8. So you have M times 68 times 68. Okay, assuming we have 68 regions of interest. So each subject, you can get this data. This is a 3D dimensional matrix, so-called tensor data. Okay, so that's the kind of weighted network we can extract. In general, if you have a data set, the data set contains not only the brain imaging, it also contains some measurement of behavior. Let's call this as let's call the connectivity matrix as A matrix. We have I to index the I subject. And let's say that we also have some measurements called YI. So our YI can measure cognition. And that's one example. Cognition is a big domain. You can have different measurements, full intelligence, memory, and so on. We can also have, let's say, sports, or the time that you do sports, right? That you just exploited. What else can Y be? The weight, the height, and some other demographic behaviors measurements. Demographic features, behavior measures we can have. So we're gonna have a pair of data for each subject. The goal is for us to establish the relationship between between A and Y. Right? We want to understand how A is predicting YI, how brain network is predicting YI. Right? So let's say we have a person, we have in ABCD data, and we have young kids, and we measure their reading ability. Okay, we measure the reading ability, and then Y is a continuous variable to describe this person's reading ability. And then we want to see if certain brain structure can facilitate the development of reading. Okay, we want to identify whether some of the elements in AI can predict YI. So this type of task. So centered around this type of task, we can develop many different algorithms. We just mentioned a tensor. Let's say we develop tensor regression where X is tensor, where A is a tensor here and Y is a scalar. Okay, A is not a network anymore. Come back to the last slide, we can say that this is our AI matrix. It has a dimension of m times 68 times 68 because we have m different features. But m could be 1 as well because maybe some features are not so informative. We're just interested in one of them, so m could be 1 as well. So if m is 1, we so this paper is describing brand subgraphs. Basically, want to select the subgraphs in A so that we can predict human traits like behavior. Okay. And also when yi, in those two cases, yi are continuous variable. And yi can be binary as well. Let's say if a person has disease or not, so y can be 0 or 1. Whether a person is really good at doing something or not, y can be 0 and 1, right? So now we have a brain network classification problem. So what we want to do is we want to extract the subgraphs so that we can classify the brain. If we can identify those subgraphs, and they can really well predict the class, maybe those, identify subgraph is really important for this classification problem. Okay. And of course, we can have some nonlinear methods. Let's say we can do auto encoding of graph value data, develop some variation autoencoders. We have graph data for A, and then we encode it into some low dimensional space, and then decode the graph data back. Develop some kind of generative model. Then you can sample in this low dimensional space and then recover this graph data. And then if you're interested in model fiber curves, those 3D objects, and we can do fiber curves modeling, and also along the fiber curve, the functional data. So there's a functional scalar regression you can do. But for today, we're going to just introduce a few simple ones. First is the tensor network characterization for analyzing the relationship between x and y. Okay, so the paper is really simple. The idea is really simple. This is one of the very high cited paper, 100 times and so on. What we try to do is, want to explore what we have is this maybe some tensor like AI and yi. So AI could be a graph or a tensor, okay? And yi is some behaviors. What we try to do is, because AI, we can stack AI together, make an X matrix where X, the first 2 dimension is the graph data, and the third one is I, and let it to AI. That is, you put your matrix like this way, right? The first 2 dimension is the graph, and then the third dimension is stacked together and get a 3D tensor. That's the X matrix. So this X belongs to n times. We still use 68 and 68 as our case. Of course, sorry. I think I should be X. I already write it. X belongs to the dimension, belongs to this dimension. V times n, and V here is 68. Okay. I just give you one example of the V. V is the number of nodes, and N is the subjects. So what we want to do is we want to do a CP decomposition. Okay, if you have heard the tensor decomposition, and CP decomposition simple is the one. What we try to do is try to estimate X as a linear combination of K rank 1 tensors. Okay, this is rank 1 tensor. So the one-to-one tensor is V out the product of V out the product of U, and V is a vector. Okay, V is a vector, is a very sparse vector. and the V after product of VK take forms like this. If you assume VK is sparse, of course we didn't assume VK is sparse. If we assume VK is sparse, what we get from those matrices is some clique graph. The clique graph means, if you have some nodes, and if those three nodes are connected, so the three nodes are connected. There's no mislines between the three nodes. And if the 5 nodes are connected, we have connections between each of them. Something like that. Okay, so you have pairwise connections between each node in your subgraph. So that's a nice thing about this VK output of VK. It just decompose the whole graph, the complicated graph into some base elements like this clique graphs. And then this UK is a weight. So it's a weight for, for n subjects. So if you look at I subject UKI, represents the weight or the score corresponding to the I subject. Okay, so UK is called subject mode. It represents, the expression of those base graphs among the n subjects. Okay, so through this really simple decomposition, compare with PCA, it has some nice properties. PCA, you have to vectorize your graph, therefore you lose the structure of the graph. However, we do the tensor decomposition, each of this VK output of VK give us something that's really meaningful in terms of graph, in terms of connectivity. So, so we're able to decompose, and the VK is subject mode and encodes the information from the subjects, therefore describe the variability among the subjects. What we can do is we can grab the first 3 K out. It's just like PCA, the first 3 K UK out. It's the first 3 PC scores, and then you can plot the first 3 scores in a dimension of 3 or R3 in the space of R3. U1, U2, U3, right? And that's where we got from this decomposition. And then what we did is we applied test-retest data set. Each subject got scanned 3 times. And then we plot the subject modes, the UK, you can understand as PCA scores into this R3 space, and then use the same symbol to denote connectivities from the same person. What's nice thing about this embedding of this plot is that you can see clustering patterns, right? Connectivities from the same person are clustered together. Right? This means that the UK can effectively extract information from the connectivity data. Just like PCA, the UK we got from this decomposition. If the networks are from the same person, they will have similar embeddings in the space of UK. Okay, so in other words, we can use those UKs to analyze our data, and UK is a low-dimensional surrogate of our original connectivity data. Okay, now we're going to use UK. We're going to display UK. This is called exploratory data analysis. When I first I developed PSE, and then I processed UK HCP data. We have 1,000 people. I was really excited. I want to see, previously people just thought that the structure connectivity does not contain so much information about cognition, about many measurements, behavior measurements. People believe that structure is more informative. So I was really curious. Let's say that let's give it a try, and I have this simple tensor decomposition let's give it a try to see if structure connectivity carries information about behavior. So how to do that? How to demonstrate that structure connectivity carries information about behavior? One simple thing we can do is we can just plot those scores, right, in a 2D space. The first U, the second U, and each dot represents one subject. And each dot represents one subject. And the spread of this represents, all the subject in this low-dimensional low-dimensional space. What's the distribution look like? And then I can color those dots according to some behavior traits. In this case, what we do is we color that using our reading, our reading score. Of course, I think I cut off the oral reading to high and low, and therefore just plot high and low. And then you can immediately see that there are kind of two groups in this plot. They're very different from each other. What does this mean? This means that structure connectivity does have the ability to predict the oral reading, right? So this is really exciting when I first saw this. I continue to do more variables. So flow intelligence, you see the same thing, the two separated clusters. Really interesting, right? And once you see this, I'm pretty convinced that structure connectivity contains information to predict those traits. So how to prove it? Really simple. Let's just develop a very simple model. I can come up with some baseline model. But they I only include age and gender in my model. And I try to predict Y. Y can be the reading ability, Y can be the substance use measurements and so on. So basically, the baseline model saying I just use age and gender and see how I can predict. And then I snap into the connectivity measurement, basically those U's and the U's. UI, let's say belongs to the dimension K. I have K of those U's from the decomposition we just did to represent the relative position of the subject in the low dimensional space. That's are my predictors. Now I have age, gender, and 5 of PC scores obtained from the tensor decomposition. What I want to do is I want to see if the second model can do better than the first model in terms of predicting the traits, right? If the second model can do better to predict the trait, it means that the new variables we add, the connectons we add, can be very helpful in terms of predicting those traits. And also we can sort those traits which can be better predicted by the second model compared with the first model. So what I do is, I draw this figure. Let's say the different color represent different features. You can just focus on the green one and focus on the green one. What I do is I sort the percentage of improvement, and then I draw the first 1, 2, 3, maybe first 10 of traits that can be better predicted by the second model. This means that the connectome is going to be related to those traits, right? Because they can better predict those traits. And surprisingly, if you look at those 5 traits which can be better predicted by structure connectivity, 5 of them are related to substance use. Okay, max strengths, frequent on 5+ drinks per day, marijuana use, all of those related to substance use. I mean, this is astonishing to me. It seems that substance use, those are young adults. I consider as normal people, normal controls. I don't think they have cognitive difference, right? In terms of cognition, there's no difference. But in terms of brain, there's something happening in the brain. Although the cognition is high level measurements, the structural connectivity is relatively low-level measurements. It's kind of like hardware supporting the software connection. Even some hardware it has some glitches, and maybe those different regions can work together. You don't really see, systematic level, condition level, there's some differences. But something is already happening with the body, and structural connectivity becomes different compared with normal controls. And also some of them related to cognition, like reading ability, vocabulary, fluent intelligence, and household income. That's pretty surprising. Okay, or somehow related to cognition as well. So while cognition also related to structural connectivity. Interesting. Okay? So very interesting finding. Okay, this is not the end of story. We want to find how different brain connectons are changing with respect to traits for the population. For example, if we say, among my population, there are some people who is really good at reading and who's not so good at reading, or who's really good at doing who's really good at reading, who is not very good at reading. I want to look at from people who are not good at reading to people who are good at reading in this low dimensional space. I want to draw an arrow, which on this arrow, when we project this data to the score, to the place, the correlation of this projection with respect to the reading is maximized. Okay, so that I can point from the low rating class to the high rating class. So I want to try to find this direction, and because this direction characterize, how the brain changes from people with low rating to people with high rating. If I can find this direction, then this direction is some low dimensional space. We're analyzing the data. You have to project them back to the original network and look at what's happening. So because we do a linear decomposition, and those U's are meaningful, and you can use the U's. Okay, those are the W, and those are VK, the basis. So you can cover this delta X, which is the change from low to high, and then plot a figure like this to see which pathway has been changed among the in the brain. And interestingly, you see that from low to high, lots of pathways get enhanced. You see the color bar, all of them are positive connections. Okay, so this means that people who has better structural connectivity in those regions tend to have better reading ability, or vice versa, because we don't know the causality in this case. We just know the association. So either this cost high reading score or high reading score, if you have like reading habit, can reshape your brain connectivity, right? So we need to collect more data to demonstrate this. And we can do this for more for drinks from low to high, right? From low to high, you got a lot of negative connections. The delta axis negative. This means that if you drink lots of alcohol, those structural connectivity pathways get reduced. And similarly, marijuana use, they have lots of marijuana use, it got reduced, and those pathways got reduced. Of course, we can easily get an intuitive feeling that more substance use is going to damage those fiber tracts, right? We got this intuitive explanation, but I want to emphasize that this analysis wouldn't make a causal conclusion. Okay, it could be that substance use caused those subtract or track reduction or connectivity reduction, or the reduction of this connectivity cause more substance use. You know, for if those person has less connectivity in those regions, maybe they have less control of their self, and they can have more substance use. So again, there's some hypothesis to test in the future. All right, I think that analysis is really interesting. I think that, you know, that's my second paper in the field of brain collective analysis. I really liked it and enjoyed it because it's the data I processed from the very beginning, and then I jump in and analyze it. Very interesting. Okay. And let's continue to do some more fancy analysis. Do we have more time? Yes. So I'm going to talk about autoencoding then, because the previous one is all linear, right? So now it's time to develop some nonlinear stuff to see if we can better predict the y, because x, a and y could have some nonlinear relationship. So the drawback of previous methods, I didn't list all of them, I just give you one example. One is linearity. It may not be sufficient to model non-Euclidean graph data because our network is graph data. And also a lack of characterization of the population distribution of networks. Basically, we cannot characterize how the networks are varying among normal population. So we want to characterize the variability or the distribution of networks, but we also want to establish some nonlinear relationship between network and behavior. That's our goal is this autoencoder. So we want to learn low-dimensional representations, just like what we did before of brain networks, and we also want to generate brain networks from our model, and we want to relate brain networks to human traits. Okay. And a few years ago, the variational autoencoder was really popular before the diffusion model, and the diffusion model now became very popular as well. The Devil E and all those image generators, the video generators, they use this diffusion model combined with transformers or fully connected networks and so on. So our goal is to learn the variation autoencoder so that we can, from AI, a graph, we can learn low-dimensional embeddings of Zi. Okay. Previously, it's linear. You know, we learn Zi or Ui in the paper. We have a linear way of learning it. Now we have a neural network to do that. So this Zi is not necessarily a linear function of AI. It would be a nonlinear function of AI. Okay. And from Zi, what we want to do is we want to generate if Xi and Xi is dominating AI. I will explain that component in a moment. And then from Zi, let's say Zi is a vector of 64, and we can just run a simple linear model to predict Yi, right? And Yi now is a nonlinear function of AI because you have nonlinearity in this layer. Makes sense? So, you know, our goal, we have three goals. We want to learn low-dimensional embeddings of our graph. We want to predict Yi, and we want to sample from Zi in the space of Zi so that we can mimic the graph and recover the graph back, connectivity graph back. Then Zi, you can assume Zi follows a Gaussian distribution of mean zero and independent diagonal covariance matrix. And then you can sample from this prior and can generate many different graph data. This is very useful in the future when we want to send some data to you. We cannot send the true data because the true data is covered by some data usage term. We cannot share it. And with this model, we can synthesize the data, just like Devil E, you know, and use this model. We can say, oh, simulate some brain graph data and then send it, you know, and then can send and share it with you. Those graph data are going to look like real graph data. So in general, we have two components. One is encoder and one is decoder in our VAE framework. The encoder is called inference network, and the decoder is called generative network. So the generative network, we have to play some tricks, and it has to be different from the existing decoder, which decodes image data. Graph data is very different from image data. Graph data is fast very naturally. If you look at the structured connectivity, I have some examples. Right, this one, if you think about this as adjacency matrix, you can see that it's very sparse. Many connections are missing, or there's no connections between many pair of nodes. So you have to be able to generate some data of AI, which is sparse. So how to generate AI sparse? A sparse AI. Right. We cannot directly use graph and output, say, 6, 8 square numbers, and then vectorize them into a matrix format. That's not going to work. So this is one of the most important tricks in our paper. That is, we try to indirectly measure AI. So this part. ZI firstly mapped to xi. Xi has a dimension of v times, let's say, p. So v means it has a v node, and p is, say, each node it has 3 features. So basically, we embed the nodes of AI into some dimension space of RP. So that when you look at the two connections between I and J, these two connections, and it's related to some kind of distribution. Okay, I have it here. Okay, for U and V nodes, and we can define the distance in this node embedding space, which belongs to RP. So each of these belongs to RP. You can look at the distance of this 2 nodes in the low dimensional space here, and the 2 distances are very close to each other, or it can be far away from each other. Okay, so the distance of the nodes gonna decide AI, and then the AI follows percent distribution with those as parameters. And we also have this gamma, which is shared by everybody, and this part is xi, which is independent for each subject. Okay, so that's the key part. And AI here is fiber count. That's why we use percent distribution. If it's not fiber count, if it's like a real number, we can use Gaussian distribution as well. But the key is that instead of directly get AI from the Z, we have an intermediate variable for xi, from Z to xi, and then to AI. The relationship between AI and Z is given by this. Any questions? So that's the decoder part. The encoder part is nothing but just fully connected a neural network. And we have to do variation autoencoder, so we have to predict the mean and variance for each ZI. So each ZI is a distribution. So there are 2 graphs, one graph predicting the mean, one graph predicting the covariance. Okay, and this epsilon represents some rate parameterization tricks. Okay. So encoder is pretty straightforward, fully connected network, but decoder utilize some graph convolutional network techniques plus Latin space graph techniques. So then let's look at the embedding space. The embedding space, again, it's very similar. So we're plotting the ZI in R3. So the ZI can be pretty high dimension, can be 64 dimensional, can be 128 dimensional, so on. But you can do SVD to the ZI, therefore you map to the lower dimension, say R3 space, and then we can still plot those dots. Again, we color those dots. Each dot represents one people with different measurements, reading, vocabulary. Okay, so you can see that they're pretty separate in this case. The reason that they're still separate compared with before is that we have y here in the energy function. So in other words, when we extract the zi, we want to extract the zi that is predictable yi. So it's called supervised feature extraction. That's why you see that those dots are very separated. This is because we incorporate information from yi in the training process. Okay, so the name I give it as supervised feature extraction. In other words, we're going to extract the features that tends to predict y better. And we're going to learn how to extract that feature using neural network. Okay, so that's why in terms of prediction, the IE gate, this is showing mean squared error, is much better than tensor PCA. This is linear regression plus tensor PCA. You can see a lot of improvement in terms of mean squared error, and compare with PCA, regular PCA. Okay. And some other methods. Okay, so because of this design, and we have achieved a better prediction in the test data set to the traits. And of course, because we have a y in our energy function, we can kind of compute the posterior probability of yi. The conditional probability of AI given yi. Okay, this allows us to sample AI for different than yi. In this case, we can sample y equal to 60 for our reading, all the way to y to 138. And you can see that the graph you samples are different. Okay, and this synthetic data allows you to look at the difference between different connectivities with different traits. This is similar to WE, where you say I'm gonna sample some graph that y equal to 138. So you sample 100 of them and take a mean. I want to sample 60. So I want to sample 100 of graphs with y equal to 60, which is reading test who has a score of 60. You can do that. Okay, and then you can compare the graph of these two and see how the graph are different from each other. And this is the difference we learn from our data. And this can help us to do inference. Okay. So I might not have enough time to finish the continuous one, but that's the discrete one. You can have a flavor, right? We have graph data. We can use lots of really interesting techniques to analyze our data, maybe to improve the analysis from simple to complex. But there are some intrinsic drawbacks when you use discrete matrices to represent brain connectivity. What are the drawbacks? The first one is that in order to form a discrete structured connectivity matrix, you have to choose some atlas, DESCAN, Distrios, MSM, some other atlas. Each of them are going to give you different results. Okay, each of them are going to give you a different connectivity matrix. And also, because you choose the atlas, therefore you choose each big brain region as one node, and you're going to not distinguish them. You're going to treat them as the same. So then information within those nodes or region of interest are totally lost. Okay, total loss. So that's information loss. So instead of treat the brain networks as a discrete graph, we can treat the brain connectivity as continuous functions. So how to do that? It's a really strange concept, but I found this is really interesting. Okay. And then we developed this service-based connectivity integration pipeline we have introduced before. And basically, we choose to analyze the connectivity on the cortical surface. And cortical surface has many nice features. For example, the cerebral cortex has a natural 2D topologist geometry. And this is the brain. You've seen this, a grown brain, a physical brain. Here, those are gray matters. It's a 2D sheet cover throughout the brain. And if you want to calculate some features, say cortical thickness, basically like how thick of your gray matter, how many neurons in the gray matter is directly related to that. And it can only be measured on the surface. If you don't do volume, it's really difficult to measure. And also, first, alignment is easier, is one-dimensional length because the intrinsic dimension of those surfaces are two-dimensional. It's not three-dimensional. Okay. And then we can use unified analysis framework for features on the surface. I wouldn't talk more about that. Okay. So from PSC, the SBCI framework, what we got is first the surface for each subject. You get your brain scan, and you're gonna extract the surface. That's your brain cortical surface, and we got the fiber tracks connecting different brain regions on the cortical surface. Okay, so the data we have now is it looks like this. So we have pair points, the beginning, the end of a streamline, and those 2 points are on the cortical surface connecting different brain regions. So we have many of these pair of points, maybe a million of them, because we have so many neurons in the brain. So, and those pair of points generate some connectivity pattern, which is something that we're really interested in. So that's what we call continuous brain connectivity. Okay, so the goal is instead of analyzing, you draw a circle, say you count how many fibers connected in this region and other regions. We say, there is a continuous procedure on the line decides the distribution of those pair of points. And that's the assumption. Okay. And then some statistical models jump in. Let's say we have to use stochastic point process on omega cross omega, where omega is the manifold space for the two cortical surface. And then we assume there's an intensity function U really decides how many end points you see between region 1 and region 2. I think this is really interesting because, it's kind of like there is a God that decides this intensity function of your brain. And then because the intensity function and different connections just get formed, you can view like this way. Or in this process of brain development, you generally have some kind of force to generate this U, and this U decides how different brain regions get connected. And this U now becomes our interest. It's called continuous connectivity because it has values between any points of regions on the cortical surface. Okay, so and then the remaining task is to analyze the U's. Similar to what we have seen before, we have to, there's 3 tasks to do. We have to estimate this U from this point cloud. The millions of pair points, we have to estimate a U. And we want to link U to behavior, say how well we can predict traits, And also we want to identify local subnetworks difference between groups. Okay, but two population which brain networks are different. Those kind of tasks we have to do. But I'm gonna skip a lot of details on how we analyze the data, just give you a sense that in terms of predicting behavior traits, this is the continuous connectivity one, and this is TMPCA. What we try to do is we try to compute the correlation between the predicted Y and the original Y in some validation or testing data set. And you can see many cases in terms of flow intelligence, self-regulation, sustained attention, executive function, so on. We can achieve much higher correlation in the test data set compared with TNPCA and some other methods. Okay, so when we represent brain connectivity as continuous functions. And in terms of hypothesis testing, let's say for a trait, you grab a set of population who has really high value for this trait, and a set of people who has lower value for this trait. And you want to see if they have population distribution difference. And this continuous connectivity actually discovers a lot of significance. Those red ones are FDR corrected significant traits. But if you use other representation of brain connectivity, you don't really see so many significance. Okay. So you have a better statistical power to tell the difference between two populations if you represent the brain as continuous connected.